So welcome everyone to the December installment of the IPM Hour. Today, we're going to hear from Fabian Menalled and Laura Berrios-Ortiz, both from Montana State University, and they're going to talk about joint management of interacting agricultural pests in small grain systems. Fabian is a professor in the Department of Land Resources and Environmental Sciences at Montana State. He received his Bachelor of Science degree from the University of Buenos Aires and a PhD from UMass Amherst. And his recent interests are really on the interaction between multiple pest species in cropping systems, and importantly, from an IPM standpoint, how to manage those multiple species, those pest issues, simultaneously when the strategies, those management strategies, the management strategy for one may affect how the different pest population grows. So that's what they're going to talk today. And Laura is also going to speak today, and she's a master's student in Fabian's lab. So I'm not sure who's starting, so either Laura or Fabian, you can go ahead and start.
I will be starting. Thank you, Matt, for the introduction. Thanks for inviting me. I want to clarify, Laura is actually a PhD candidate. So she got her master's from University of Puerto Rico, and we were extremely lucky that we were able to recruit her for this project. So, and she's been doing a fantastic job. And thank you everyone for joining us. And what I want to do today is to share with you some of the research we've been conducting for the last several years. The first half will be a project that was run by Eli Karen. He was at that time a PhD candidate in my lab. He was actually a student from the stats department. And then I will be shifting to the research that Laura is currently doing. So one is research that we finished, and the other one is a researching project, and we'll show a preliminary research of results that we are obtaining with Laura. But like Matt was saying, what would be interesting in trying to see what can we do to manage multiple pests when they interact with each other. So I'm going to be sharing my screen, and I think you can see now my presentation, correct?
Yes.
Perfect, thank you. So the issue here is that sometimes when we have, most of the time, I would say most of the time, we don't have just one pest group at a time, either weeds or pathogens or insects. They tend to occur together. Now, what happens is that, and I think the first question we want to ask is, why do we need to consider these multiple pest interaction, these multiple trophic interactions, when we're going to be making pest management recommendations? And I think the issue is that those of us who have a sort of gray hair is that when we were undergraduates or grad students, we were trained to think at one problem at a time, this idea of what I like to call sort of isolated silos of knowledge, one issue at a time. And if we are looking at pests, then we may be interested in looking at weeds, and we have weed scientists working in one department, or entomologists working in another department, or plant pathologists working sometimes even in a third department. So they don't really talk to each other, and they come with their prescriptions. And I like these photos, which are really old silos. And if I'm a wheat scientist and I look at this wheat field, and if it is a winter wheat, I will probably be concerned here in Montana about downy brome or cheatgrass. That's Bromus tectorum. Is a winter annual weed, and I'm pretty sure that most of you know it because it's not just in Montana, it's in the whole western region, not just in croplands, but also in rangelands and wildlands. Now, as a wheat scientist, and if I have the traditional wheat fallow system that dominates Montana, my recommendations will be to use glyphosate during the following year, or maybe preceding, and then use some of the different herbicides we have for in-crop. Now, you probably know where I'm going. The overuse of those herbicides basically resulted in herbicide resistance, and we have currently in Montana cases of resistance to group 1 and group 2 herbicides, the ALS and the ACCA inhibitor herbicides, and only a few miles north of here in Lethbridge in Canada, they already found Bromus tectorum biotypes which are resistant to group 9, the EPSP inhibitor herbicides that would be glyphosate. So that's a really scary situation because we are basically losing the tools that we have to manage cheatgrass both in crops and the fallow year. And because of that, wheat scientists have been considering some cultural practices management, such as, for example, increasing the seeding rate or modifying the fertilization regime or using more competitive varieties, which is good. But if I now use my entomology hat and I look at this field from an entomology perspective, I will probably be concerned on the wisdom sawfly, which is the most damaging insect pest in our grassy crops in wheat. For those of you who don't know, the wisdom sawfly has one life cycle per year. It's a short-lived insect. The adults, female adults, fly for a few days, a couple of weeks perhaps, and they oviposit on the best stems. Then what happens is the larvae will burrow through the stems, and that in itself creates around 20% yield loss. But what happens is they create a diapause chamber, and in itself it's not a major problem, but the issue is that then the stems break and there's a lodging of the mature wheat stems, which adds also in the decrease of yield and also some major problems to harvest that wheat. Now, if I now shift gears and I use my plant pathology hat, I will probably be concerned about a Fusarium, maybe the Fusarium head blight or the Fusarium crown root, which are also very commonly found here in Montana. Now, the issue is that, as I said at the beginning, these pest groups don't occur in isolation, and they actually interact with one another. And for example, the wheat stem sawfly actually prefers to oviposit on grassy weeds versus wheat. So they will oviposit on cheatgrass. If they oviposit on cheatgrass, they can survive and produce new individuals. If they oviposit, for example, in wild oat, they will die. Cheatgrass is also a host for Fusarium. So we can think that Fusarium is a biocontrol agent for cheatgrass, but I'm sure that farmers will hate to think of that way of, using Fusarium to control our cheatgrass. But also, the Fusarium will kill the larvae that is overwintering within the wheat stems. So you can see there's all of these direct interactions, but we can also think there will be some indirect interactions. For example, if we now have a cheatgrass that is infested with the Fusarium, the quality of that cheatgrass is going to be decreased, and cheatgrass and the wheat stem sawfly prefers to oviposit in really large stems, so they may not like the cheatgrass, and so on. So there's all these complex interactions that we don't consider. These are biological and ecological interactions. We then move into our management. For example, in the case of the wheat stem sawfly, there is not a really good viable chemical option available in the market. There's been a few products released, but not, farmers are still relying on cultivar selection. Here in Montana, what they tend to do is to use solid stem varieties. So the larvae will not be able to burrow, and they basically died. But those solid stem varieties are not very competitive. So if you have a weed, you may not be able to use those solid stem varieties. So what you may do is to increase your seeding rate to control those weeds, that cheatgrass. That's good for the weed management, but unfortunately, when you increase your seeding rate, what you do is you lose the solidness of the stem. So it's good for the weed, but it's not good for the insect. And also, as you increase the seeding rate, not only you lose the solidness of the stem, there's going to be more moisture challenges, particularly in places like Montana, where we don't receive a lot of moistures, and that may be a problem in terms of a pest management or the pathogen management. So we have all these management contradictions, and we can think like we have a big complex problems, and if we try to tackle it with a simple solution, that simple solution is probably going to be clear, simple, but wrong. And this is because, like I tried to explain, we have all these direct and indirect interactions, and sometimes those management practices could be backfiring on us. So this is basically the framework of today's presentations. As I said, I will be showing two studies. I will be showing the first one, the one that was done by Eli, where he was working on the interactions between the wheatstem sawfly and cheatgrass, and then Laura is going to be talking about the interactions between cheatgrass and Fusarium. So let's start with the research that was done by Eli Karen. So we conducted this study at 3 locations within Montana, sort of in a north-south gradient. And in each one of those 3 locations, what we had is 3 reps, and the experiment follow a split plot design. First, we have 3 wheat varieties, and we're representing them by the different colors. A sawfly resistant variety, a solid stem. Then we have a drought tolerant variety that is really good in terms of competitions against the cheatgrass. And then we can call it a wimpy variety, a high yield variety, but is not competitive and doesn't do good under the presence of the wheatstem sawfly. And we seeded them at 3 different seeding rate. A low, which is the recommended seeding rate, then a medium and a very high seeding rate. Again, that could be good for the weed management, but not that good for the sawfly and pathogen. And we were relying on the existing pests for both the wheatstem sawfly and the cheatgrass. And in order to create a range of densities of Bromus tectorum, we applied herbicides at a very low rate, 0.2 of the recommended rate. So we expect to see a lot of cheatgrass, 0.4 or 0.8. So we were expecting to see just a few scapes. And inside each one of these split plots, we have 5 rings. You can see 3 black rings and 2 green rings. And basically, we use those 2 rings to measure productivity early in the season, and then the other 3 rings to take them to yield. So this is a photo of one of our field sites. You can see 2 reps, and then 1 seeding, 1 crop variety at the seeding rate, and they were again split by the different herbicide rates. So we were applying the herbicides, and then we have these rings that I mentioned. 2 of those rings were harvested early in the growing season, and we checked for the biomass of cheatgrass. And then in the other 3 rings, we took them to maturity. We harvest again cheatgrass, check for their biomass, seed production, and also we harvest all the wheat. So we brought them to the lab, separated the cheatgrass from the wheat, and then for each one of those stems, we carefully cut them and check for the presence of the larvae of the wheatstem sawfly. And then we check for the yield of each individual plant. Now, we have a lot of data, which was pretty complex to analyze, and that was really good and handy that Eli was a student from the stats department. So what he decided to do is to use a structural equation modeling approach. And basically, if you're not familiar with this approach to analyze data, what you do is you establish all the potential relationship between the variables and then solve those equations simultaneously. This is an example that I'm showing here for a paper that was published 2011 in the Canadian Journal of Plant Science, and they used this structural equation modeling, or SEM, to explore the yield of oat. And you can see in this diagram that they explain the yield as a function of the seeding density, the actual or the target seeding density, the actual seeding density, the panicle density, the seed size, etc. And they were able to predict the yield. So basically, they solved those equations simultaneously and found all these different interactions with the different R-square, positive or negative. And that will be just with the variables they measured, but you can also include those indirect variables or latent variables, and this will be the model they develop. So it's a relatively complex model, but allows you to predict yield as a function of different independent variables, all considered at the same time. So this is the model that Eli developed. Basically, we have management, and for management, I'm talking again, the 3 crop varieties that we use, the density of the wheatstem sawfly, WSS, and the abundance and density biomass of the cheatgrass or downy brome, and the yield, and their direct interactions, those will be the dotted lines in black, as well as the indirect interactions, that will be the brown curved lines. And Eli established these equations that I'm showing here. Again, he was a student of the stats department, who was really savvy in doing this, and established those relationships between the indirect variables. And then he solved that model, the set of structures using Bayesian inference. If you, like myself, were trained as a frequentist analysis in stats, usually what we do is we collect data, and then we check for the probability of your data set. You basically have this sort of normal distribution, and then you check for the probability of that data set to be true following a certain hypothesis. So you run your ANOVA test or your t-test, and then you reject or fail to reject the null hypothesis. In the case of Bayesian stats, it's a little bit of a opposite direction of the logic. You have a priori knowledge, for example, the behavior of the wheatstem sawfly in the presence of cheatgrass, or the relationship between yield, wheat yields, and cheatgrass abundance, and you basically establish your a priority or your prior probability, that will be the yellow line. You collect data and you run your model and you establish a posterior probability. And the narrower that bell shape means you are basically increasing the probability of the hypothesis to be true. So basically you test your hypothesis based on your data. So I will be showing results with this type of bell-shaped curve. The more narrow they are, the more precision we have or more confident we have that the hypothesis is correct. The good thing of Bayesian stats is it allows to incorporate those pre-existing knowledge and also these indirect and direct interactions, and also is relatively powerful with low data numbers. Well, I mean, we usually, like I said, we only have 3 fields, 3 reps, so our replication is not extremely large. And usually, if you have more frequency stats, you will have not too much power in your tests. So this is an example of how we incorporate our prior knowledge or pre-existing knowledge. We knew that the Whitsome sawfly likes to oviposit on relatively larger stems. So if there's a lot of competition, the stems will not be that large, and the Whitsome sawfly may not like it that much. And also we knew that if there's cheatgrass or Bromus tectorum present in the field, the Whitsome sawfly will actually prefer to oviposit on those Whitsome sawfly, on those, sorry, cheatgrass stems. So what we can think that we'll have a frequency of distributions of grain yields per individual head, and it will probably follow a normal distribution with your mean and your standard deviation. And that will be, say, a situation where you have no cheatgrass. So the expectation will be a normal distribution. And with stem sawfly, we know they like large stems. So there should be a minimum size of the stems under which they don't oviposit anymore. They are too small, okay? So we can predict that there will be a certain mean yield per individual stems in the absence of competition. Now, if we have cheatgrass, there will be competition between the cheatgrass and the wheat. So we expect that the wheat will actually be yielding less. We will have more smaller stems. And there's 2 options. It could be that the Whitsome sawfly will not change its behavior and still have that minimum size of the stem under which it will never oviposit in smaller stems. And if that's the case, then because they have that threshold fix, we will have more or less the same mean yield of those uninfested stems. But if the Whitsome sawfly change the behavior, and because it's now in an environment where we have more and more smaller stems due to competition, it is possible that the Whitsome sawfly will start to choose to oviposit in smaller stems. So we actually collected the stems, we knew the density, we knew their yields, and we start to assess those potential changes in the behavior. And this is a little bit of a busy graph. I'm going to try to explain it to you. In the x-axis, we have the mean yield per head of the wheat, and we have the 3 seeding rate, low in the bottom, medium, and high seeding rate. And then we have the 2 varieties, the sawfly tolerant variety, solid stems, and then we have the drought tolerant varieties, okay? And then you have these different colors. Dark and black is basically where we spray 0.8 of the herbicide rate, so very low weed pressure, not too many cheatgrass. Then 0.4, sort of an intermediate abundance of cheatgrass we saw in the field, and then 0.2, basically we have quite a lot of cheatgrass infesting those fields. And those graphs basically represent the those bell-shaped curves by looking from the top, okay? And what we see here is that in 5 of the 6 cases, we actually saw a movement of the mean yield of the uninfested stem. So that's telling us that in most of the cases, the Whitsome sawfly will actually change their behavior and start infesting smaller stems if there is cheatgrass. So you can see here, this is will be an example of a biological interaction that we usually don't consider between an insect and a plant. And it's not our crop, there will be a weed as a function of the seeding rate and the variety of the crop. But we are concerned about management, and the question is, what will be our management recommendation? So in the situation of a weed-free environment, trying to move my careful. What I'm presenting here is the posterior density of the yields. So how much yield are you going to be getting for the drought-tolerant variety in the top and the sawfly-tolerant variety? So in a very high herbicide rate, 0.8, there were almost no Bromus tectorum. And what you see here is that, as expected, because we have quite a lot of widths and sawfly in that situation, the best situation was to seed the sawfly tolerant variety, of course, at a low seeding rate, because if you seed at a high seeding rate, you lose that solidness of the stem. And that was much better than using a drought tolerant variety. So for one pest, when you only have width stem sawfly, then use the management, the recommended management for that pest. But the next question will be, what happens if we all of a sudden increase our weed pressure and say that would be a situation where we apply 0.4 of the herbicide rate? We could think that if we think in those isolated silos, we say, okay, we'll use the sawfly tolerant variety because that's good for the width stem sawfly, and I'm going to increase a little bit the seeding density, so I will get the control of the Bromus tectorum. But as I mentioned, you start losing the solidness of the stem, and that's basically what we saw. And now you see that the best option is not to increase seeding rate and use the sawfly tolerant variety. You lose that solidness of the stem, but the best option was to go for the high seeding rate of the drought tolerant variety. So in other words, what I'm saying, when you have two pests co-occurring, a weed and an insect, in this case, our recommendation is go for the high seeding rate of the competitive variety, take care of those weeds. Again, assuming you don't have herbicide because of herbicide resistance, take care of those weeds, reduce the seed bank, and once you have that sort of monster under control, then shift your management practice and start controlling the width and sawfly. So unfortunately, our results are confirming that what we thought in advance, that we cannot manage those two pests together due to those interactions. And that's basically telling us about trade-off. You have to understand that in this scenario, we could not find the best management for both pests together. So what will be the recommendation for an extremely weedy environment? And what we saw here is that our results were really, really bad. We were getting very low yield, and basically that was regardless of the variety that we seeded or the seeding rate. So in other words, what we can say that in these small grain systems, weeds are the driving force in terms of our weed management decisions. So going back to the initial question, is it possible to develop a joint with some sawfly cheatgrass management program? Yes, but we need to do that not in isolation. We need to understand that those two pests interact with one each other. There's this different behavior changes in changing the wisdom sawfly behavior under the presence of cheatgrass. There's all these indirect interactions. And because of that, we need to understand there's going to be trade-offs. And if you're sort of interested in this research, this is a paper that we published a few years ago with Hilary Kern, was published in PLOS One. You can contact me and I will be glad to share with you a copy of this paper. So that's basically the first half of the story. And with that, I'm going to be shifting gears, shifting the presentation to Laura, and she is going to be showing her current research.
Hi, everyone. Thank you for the opportunity to be here and the introduction. I want to paraphrase my stats professor, Dr. Borkowski, when he mentioned in class that researchers may shy away from these type of studies because of its complexity, and nonetheless they are. They take a lot of resources, work, and time, but they are also important to address how to tackle complex issues. This is not to undermine organismic approach to research. It's absolutely essential and it explains many reasons for these types of interactions, but to make recommendations that take into consideration these interactions, then we need to broaden the view to see all the components of that particular issue. Fabian, I can't change the, thank you. So we know that there are multitrophic interactions in nature and in agricultural systems are no different. Pests interact with each other and management inputs affect these interactions and they affect how crops respond to them. In this case, we're looking at Bromus tectorum and wheat like Eli did, but we're looking at our plant disease is a wheat crown rot, which is caused primarily by Fusarium pseudogramminearum. And we're also looking at resource availability, in this case nitrogen, and crop density by seeding rate. Our study objectives are to understand the impact of resource availability, nitrogen, disease pressure, and seeding rate on Bromus tectorum and wheat competitive interactions, to assess Bromus tectorum demography and population dynamics, to evaluate wheat crown rot pathogens, and to develop a decision-making management tool that takes into consideration the interactions and the particular economic goals. Our test sites are the Northwestern Agricultural Research Center in Kalispell, the Southern Agricultural Center in Huntley, and the Post Agronomy Farm. At each of these sites, the experimental design was a randomized split plot study with 4 repetitions in Huntley and 5 repetitions in Kalispell and Bozeman each. The main plots were randomly assigned to one of 2 winter wheat seeding rates, high or low, and subplots were assigned to a factorial combination of present or absent Bromus tectorum, high or low nitrogen levels, and Fusarium pseudogramminearum protection by having half of the wheat seeds treated with fungicide for a total of 16 treatment levels per rep. Soil samples were taken throughout the test sites before planting to evaluate nitrogen and then fertilized at an appropriate rate for the low at 87 kilograms per hectare or at a high at 200 kilograms per hectare. A month later, soil samples were taken in each split plot and sent to evaluate nitrogen, phosphorus, potassium, and organic matter. Then seeding rates were low at a rate of 67 kilograms per hectare or high at a rate of 101 kilograms per hectare. Bromus tectorum was seeded at a rate of 500 seeds per square meter in those split plots that had the presence of it, and it was incorporated by rake. Then disease pressure was applied on all of the field by inoculating at a rate of 500 sterile oat seeds with Fusarium pseudogrammineatum and a rotary hoe at 8 centimeters deep. And with this, we'll create a life history model for Bromus tectorum by analyzing population dynamics and demography. Each split plot that shows flags, which would be half of the whole field, had Bromus tectorum, and each of these had a total of 8 rings. 4 large rings for the demography and 4 small rings for the population dynamics. Each ring was harvested and the biomass was weighed for Bromus tectorum. Then we'll also be looking at seed production and seed survival, where we have 4 buried seed bags that we dig up each season and plant in germination paper to look at the after fall or after winter survival. And then we're also looking at seed predation by placing seed cards in the field and removing them 48 hours later. Now, all this information will let us build a Bromus tectorum population dynamics model where we're looking at the seedling recruitment, how many seeds were produced, how many survived, and how many seeds, how many adults actually produced flowering tillers. Then during mid-season, 5 wheat samples per split plot were taken to the lab, and we isolated fungi colonies, counting them from the root sections. We also have samples that are dry that will be looking at the molecular composition in the crown for those diseases. So far, we've seen Fusarium pseudogrammineatum. We've also seen 2 types of Cochleobolus and other types of Fusarium as well. Then at harvest, we're looking at yield, and also we're looking at quality through dockage, protein content, moisture content, and test weight. And then with all of these components and how they're interacting, we'll be able to develop the decision-making tool where the producers will have available and they can decide where do they want their most efforts to go. Now that they have that understanding of how those interact, then they can make the best decisions for themselves.
So before we break and go into the the questions, I'm pretty sure that someone is going to ask us, okay, you talk about the Whitson sawfly and cheatgrass, between cheatgrass and Fusarium, but can we actually put the 3 pest groups together and try to assess or develop a pest management program, that consider all these different pest groups at the same time, and also considering their indirect interactions? And I think it is possible, but it's not going to be simple. And this is because the inferential space we usually do in these really large studies, it takes a lot of work to do these studies in the field. The plots are relatively large, but still we have a lot of variability. So it's not impossible from a research point of view. It is very challenging, and we definitely will welcome the opportunity to work with other people. It will require a lot of effort, but I think it should be, in my personal opinion, sort of the next frontiers where we should be going in terms of pest management. So in essence, what I think is that when we look at these sort of ecosystems, we should understand they are not just complex. Agroecosystems are even way more complex than we can think, and we need to start rethinking our approach to study and manage them. And with that, Matt and Steve, do we have time for questions.
Oh, absolutely. Thank you, Fabian and Laura. I'm looking at the chat currently to see if we have any questions in the chat. So for our audience members, if you would like to, you can either type your questions in the chat. I don't think we have a question and answer section, so just type your answers, your questions in the chat, or the other possibility is you can take your microphone off mute and simply pose your question that way. Or you can raise your hand too. Any way you would like to do it is fine. So we have about, actually we have about 15 minutes for questions, so we're in good shape. So if I don't hear a question here relatively soon, I'm going to go ahead and pose my question. I'm not currently seeing any questions. So I'll go ahead and ask mine. It was pretty clear that in the experiments that you could manipulate weed pressure by adjusting the rates of herbicide use. But it looked like you were relying largely on natural infestations of Fusarium and wheat stem sawfly in your studies. So on sort of a scale from 1 to 10, where 10 is high pressure and 1 would be fairly low pressure, how high was the pressure from wheat stem sawfly and Fusarium in these trials?
So in the case of Eli, we were working with some sawfly and cheatgrass, and pressures were very high. I would say the scale of 1 to 10, 9 for cheatgrass. Definitely when we did not spray, or we have the 0.2 herbicide rate, which is almost nothing, it was overwhelmed the system by the cheatgrass. We actually asked farmers to give us really what fields, saying fields that they knew that they have cheatgrass, and we were working in areas where we knew there was with some sawfly around. It's not that simple to manipulate density of with some sawfly. So we were relying on existing population of with some sawfly. And on the scale to 1 to 10, you may need to ask David Weaver, who was the entomologist specialist here. My gut feeling will be in the range of 5 to 7, 4 to 6. So we have a pretty good infestation of with some sawfly. We actually wanted to, in that original project with Eli, wanted to also, we were monitoring Fusarium, but we didn't really get a good coverage of Fusarium. We have areas, we have a lot of Fusarium and a lot of zeros. And we said, that's what I was talking at the very end. It really requires a way larger inferential space. And this is why, in the case of Laura, where we are now working in research farms, we work in plots that we know there's very little to no cheatgrass, and these are MSU farms, and they let us put a lot of cheatgrass seeds, Bromus tectorum seeds, and we actually put in the inoculum of the Fusarium in all the fields, and then we seed with and without the fungicide. So we are actually fully manipulating the presence of those factors. And Laura, you can answer me my answer. I mean, we saw cheatgrass was overwhelming against, and how much Fusarium do you think you will put from 1 to 10?
Well, we certainly have been able to recover Fusarium from the root section samples that we took in mid-season. We still haven't analyzed for the qPCR, but I'm sure that we'll find a lot of Fusarium pseudogrammineatum, including those probably that had fungicide initially. We'll just have to see if there was an effect on how deficiency of the fungicide under all those components.
Okay. So we do have a question in the chat, and the question is, it sounds like these trials were put out on research farms. Is that correct? No.
No, actually, the first study, the live study, was done on farmers' field.
Yeah.
So there were producer fields, and the second study, yes, it is done currently. It's the one we're currently done, if Laura blurs his back, he will see a lab full of bags that we're still analyzing. So we're in the process of we are processing those samples. Those were done in research centers. We have 3 locations, and I would say they're pretty representative. You can see all the bags we have. So
oh, nice.
I think they're relatively well representative of farmers' field. We don't need, we repeat the same management practices that a farmer will do.
So since you had studies in both a research farm situation as well as a producer farm situation, understanding that they were different studies, could you make the statement that the studies at the research farms adequately predict what would happen at a producer field, or do you think there are differences there, just in, well, a variety of different factors that could affect the results?
And I see Mary Burroughs, he's here. She also works in the same experimental research station that we work. I think really well representative. We particularly work in the weed science component of the farm, and we have a lot of weeds. If we were going to be doing where the breeders are working, that's weed free, and they don't, they want to get really good, tests for their varieties. My feeling is that it is representative of farmer field. We use farm equipment and similar techniques that we use.
Yeah. Okay. Laura, anything to add?
I just maybe comment if there are producers who are willing to let us inoculate their fields with Fusarium and plant cheatgrass. I mean, I guess these types of study could be done, but I think as far as Eli's study, it shows that yes, you can still look at those interactions. We follow the same usual weed producing protocols, so I think, yeah.
Yeah. So we have another question, as Fabian mentioned. So the question is, whoops, it's scrolling around. You talked about pesticide resistance in general. I think you mentioned specifically herbicide resistance at length early in the talk. And could you talk more about how this management paradigm fits into the alternative options producers are evaluating to address pest and climate stresses? And the climate stresses, could be drought.
That's a great question. As I said, we're in the end of the semester. We just finished teaching weed ecology and management, and I think the last lecture was on that topic, basically. There is a paper that was published last year. There is the prediction of weed science in the year 2050. And there's a bunch of weed scientists and people from the industry that put that paper together. And they basically said that at the rate of discovery of new pesticides that's flat since the last 30, more 40 years, and the rate of increasing of herbicide resistance, at least, they predict that we will be running out of options of chemically based management options by the year 2050, which is, I think, is a pretty good prediction. We already have biotypes in Montana of wild oat which cannot be controlled with any in-crop herbicides. They have multiple resistance. Like I said, we have glyphosate resistant and ALS, ACCA resistant in cheatgrass, not multiple resistance, luckily, yet. We have kochia with multiple herbicide resistance, including glyphosate. So yeah, we are running out of options. And if on the top of that, you put that at least for some herbicide groups, and I will be using my herbicide, my weed science hat now. In terms of weed management from a traditional chemically based approach, stomata will be closed when you have high CO2, thicker cuticles, when you have moisture challenges. So we are seeing that the performance of herbicides decreases under the predicted scenarios. We are running right now a study in the growth chamber with increased CO2, and we're trying to see the critical period with control between common lamb's quarter and carrots. And those common lamb's quarter are growing like garden under the high CO2. So it is scary. It is scary. I think we are getting two monsters coming at the same time. The challenges that climate change is imposing us, and the issue that we are losing those chemically based options. So only I can say that, lucky for me, I will be retired before the year 2050. Sorry, I will be dead. That's good.
Well, thanks for that uplifting note, Fabian. So we do have another question. So how have growers reacted to the findings so far, especially to the idea of complexity of the idea of the complexity of the interactions? Do they think that this is something that they can use in management, or is it a throw up your hands kind of situation?
No, I first of all, I could have given had I knew this introduction, I could have given a different, this question, I could have started with a different introduction. I have a question, I have a slide, sorry, of a farmer looking at a field that looks yellow, and it's like they are integrating all together these questions of like, why is it yellowing the way it is? It's a nutrient deficiency, it is a pathogen, will I get weeds because it's a crop? So they are actually integrating these problems at the same, in their brain, and the same question will be for a small scale grower. I mean, is this weed providing a source of pollen for a pollinator or a parasitoid? It's okay, can I tolerate it? It's going to bring a pathogen. Farmers in itself are integrators. So we are not showing anything new to them. I think it's newer for us in academia than for farmers. So my experience when I show these, they go like, oh yeah, that makes sense. It is complex, we knew it, but they are integrated.
Yep, makes sense. I'm not seeing any more questions in the chat. Does anybody have their hand up? I don't see any hands up.
I just like to add something that kind of meshes both of those questions together. Farmers know more about what's happening in the field, in their practice, in their farms. Every day they're there. So how this fits into those alternative options is to also make extensionists aware so that they can make their best recommendations and give the best options to the farmers so that they can make their own decisions as to how they handle all the systems there in their fields.
Great. Thanks, Laura. Right. So I think that may do it for us. Steve, I think you might have information about the January IPM hour coming up here.
The January IPM hour will be on Wednesday, January 11th. It does not conflict with any World Cup action at all. It'll be at noon Pacific, and Judith Barrasso from Montana State will be our speaker. And and the topic and the rest of it will be in the newsletter coming out on January 4th.
Outstanding. Thank you, everyone, for joining us for the IPM Hour, for the December installment of the IPM Hour, and enjoy your holidays.